right, thank you very much, Olga. It's an absolute pleasure to be here, and thank you for everyone that's joining online. I also want to thank Odmr for this great initiative. I've really enjoyed watching these presentations over the past 2 years, and given the current challenges with the pandemic, it's been great to be able to follow the research that's happening to do with diamond technology all around the world. So thanks very much for organising that, and it's a pleasure to get up a little bit earlier to give the talk today. So what I thought I'd tell you about is some of the work that's happening here at the University of Melbourne in the areas to do with using diamond quantum sensors for probing interesting properties to do with biological systems. And so two of the more interesting systems I'll tell you about in today's presentation are looking at the teeth of this little animal here. So this animal is referred to as a chiton and has very interesting and amazing teeth that I'll tell you about. And we're studying the magnetic properties of their teeth using these diamond sensors. And then I'll also talk about some more recent work that we've completed looking at the magnetic properties of iron particles, which have been found within the inner ears of these homing pigeons. And so I hope you'll see by the end of the presentation, I think there are lots of opportunities and interesting applications to do with diamond materials, whether it be these single crystal diamond materials or nanodiamonds. I think our group is interested in using both types of materials for a range of applications, and I'll talk more about that as we go on. Before I start and get into the science, I just wanted to highlight how much activity there is around diamond technology and quantum sensing here in Australia. So here on the map, I've listed all of the universities that I know about that have leading academics with research programs related to diamond research. And you can see that there's a lot of activity, including from CSIRO, which is a government organisation here in Australia. And so we've got representation from South Australia. Victoria has a strong history in diamond related research. New South Wales is also growing in stature, and there are also key researchers in the University of Queensland. So there's a lot happening over here, and a lot of the community is very close and working together with one another. In addition to that, we have a number of ARC centres of excellence, and in particular, there's a Centre of Excellence for Quantum Computation and Communication Technology that has a diamond research program embedded into that. There's also FLEET, which is the Centre of Excellence for Future Low Energy Electronics and Technologies. And there's also a separate diamond focused program within FLEET. And in addition to that, we're now starting to see commercial diamond startup companies. So two I've named here are two of the more prominent ones. So there's a company referred to as Quantum Brilliance you may have heard of. So Quantum Brilliance are trying to use diamond materials to build room temperature quantum computers. And then there's Carbon Cybernetics, who are using ultra nanocrystalline materials to create stimulation probes for neurological applications. And there are many more startup companies that are on the horizon here in Australia. So I just wanted to highlight the history and interest in these sorts of materials that's going on down here. Now, in terms of here at the University of Melbourne, academics and researchers here have been looking and exploring diamonds for well over 20 years now, and we've established an impressive set of facilities for the characterization, fabrication of diamond materials, as well as understanding the theoretical properties. So in particular, we've got a number of CVD diamond reactors. So we have two at the moment. We've got another one which will be installed in the coming month. This will be the first diamond reactor that will be able to produce isotopically pure diamond carbon-12 diamond material. So we're really excited about the prospects that that will bring in terms of these sensing applications that I'll talk about throughout the presentation today. We've also got a range of nanodiamond processing facilities, annealing furnaces. We do a lot of work with chemical functionalization of surfaces of diamond. We've got clean room and nanolithography systems to start to do device fabrication and prototyping and these sorts of things. In terms of the characterization side, we've built up a strong track record in microscopy and imaging NV systems in particular. and NV defects within diamond. We've got 3 confocal microscopes, 5 wide field microscopes, and 2 live cell imaging wide field microscopes, which are being used routinely to do a lot of these biological demonstrations. More recently, we have installed a wide field quantum magnetic microscope, which allows us to cool materials down to temperatures down to 4 Kelvin. And also we can do studies in the presence of external magnetic fields up to a Tesla. So this has been a really interesting and important piece of infrastructure. And I think you heard from Jean-Philippe Tétien about some of his results that he's been exploring with these sorts of systems. In addition to that, we've got a strong theoretical team, which has been led by Lloyd Hollenberg and Liam Hall. And they've got tremendous expertise in spin bath physics, looking at new protocols for NMR and EPR spectroscopy. They do a lot of work with hyperpolarization. And we also do a lot of work in the photophysics modeling of defects in diamond. And we're starting to get into a little bit of machine learning. So we're quite broad in terms of our interests in diamond materials, and our focus is really now turning towards some of the applications of these materials. So in terms of the system that we're working with, the workhorse behind a lot of the science that's happening here at the University of Melbourne is the Nitrogen Vacancy Centre in diamond. And I'm sure everyone online will be aware of this wonderful defect and what it can do. So the Nitrogen Vacancy Centre is formed when we replace one of the carbon atoms in the diamond lattice with the nitrogen atom, and we have an adjacent vacancy. Now this defect can can occupy 3 different charge states. So it can be a plus, a neutral, and a negatively charged state. It's the negatively charged state that we're interested in for a lot of these quantum sensing applications. Although there are a lot of interesting physics that's happening with these different charge states as well. But for sensing and the purposes of this talk, I'm going to refer to the negatively charged state of the NV as NV from this point on. And so what I thought I'd tell you about are 2 of the interesting sensing protocols that have been employed using this defect. And these sensing protocols have allowed us to start to detect interesting properties to do with magnetic fields and temperature and a whole range of other properties, which I'll touch on shortly. Now, the first sensing protocol that's often used with these sorts of defects is something called optically detected magnetic resonance. And so the NV center, the negatively charged NV center, has some really unique properties that allow things like Odmr to be done. And so the first unique property is the fact that if we shine green light onto these materials, we can polarize the electron spin into this MS equals 0 state. And this is one of the really unique features of diamond and the NV center in particular. So we get spin polarization for free, if you like, by just shining green light onto this system. We can then manipulate the electron spin between these ground state spin levels. So we can manipulate the spin from the MS equals 0 to the plus or the -1 state, simply by applying a microwave field at the appropriate frequency, which is the energy difference between these 2 levels, which is typically 2.87 gigahertz. And so the protocol for monitoring the position of these energy levels is pretty simple. We just monitor the intensity of the light that's coming back from the NV center as a function of microwave frequency. And when we transition from the 0 to the plus or the -1 state, we typically see less fluorescence from these plus and -1 spin states. And so what you'll see is when you make that transition, the fluorescence intensity will decrease. And when we make the transition to the plus state, we get another decrease. And so this allows us to precisely map the position of these energy levels very accurately using single NV centers, or we can use ensembles of NV centers. It doesn't really matter. The principle is the same. Now, this optically detected magnetic resonance spectrum is really useful. So if we have a local static magnetic field nearby, so if I come along with a little bar magnet, for example, what happens is these energy levels will split. They will Zeeman split. And so if we run our protocol again, we can measure the amount of splitting, and using the gyromagnetic ratio, we can infer the strength of that nearby magnetic field. And so Odmr is really useful for detecting static magnetic fields. And I'll show you some examples of that in the imaging section to come later on. The other really useful sensing mechanism that we take advantage of is something referred to as the spin lattice relaxation time. And this refers to basically the spin lifetime of the NV. And so, as I said before, if we're showing green light onto the system, we can polarize the NV spin into the MS equals 0 state. If we turn that laser off, that spin will then start to interact with neighboring spins within the diamond, but it will also interact with environmental spins that are outside the diamond. And so what will happen is that spin will relax to a mixture of these three states. And so you'll start off with some strong intensity when the electron spin is in the MS equals 0 state. And as we increase the dark time or the periods between the optical pulses, we'll see that spin relax. And that'll have a characteristic time associated with that, which is referred to as the T1 relaxation time. So the 1 on E time is that T1 relaxation time. Now, the T1 time is a really useful mechanism for detecting whether there are any fluctuating magnetic field sources nearby the NV. And so in particular, if there are any magnetic fields that are fluctuating on the order of 3 gigahertz, that will significantly affect this relaxation time. And so by looking at the change in the relaxation time, we can infer whether there are fluctuating sources of magnetic fields present. So these are the two main sensing schemes that we have worked with and that the field is looking into. There are several others such as spin echo and CPMG and these sorts of things which you may have heard about as well. And so these sensing protocols have allowed us to start to explore very interesting applications to do with these sorts of systems. So well over a decade ago now, we started to explore the idea of trying to get nanodiamonds into biological systems. And so we were the first to show that we could actually do optically detected magnetic resonance in a living cell. And so in this work, we showed that we could track a nanodiamond containing a single NV center, measure its Odmr spectrum, and you can actually infer rotation that's going on with these particles within living cells. Following that, we started to explore the idea of using T1 for detecting fluctuating magnetic fields and showed that with 15 nanometer nanodiamonds, we could embed those into artificial cell membranes that were labeled with spin labels. So in this work, we used gadolinium, for example. And using a single NV and a single nanodiamond, we were able to detect the presence of 2 or 3 atomic spin labels in these artificial cell membranes. And so that was a really interesting demonstration of the sensitivity of these sorts of systems, getting down to near single electron spins. And then later that year, Misha Lukin's group came out with this beautiful nanoscale thermometry paper and showed that we could use the position of these energy levels to infer temperature as well as magnetic fields. So temperature has the effect of shifting these plus or minus 1 energy states rather than Zeeman splitting them. So you can discern a difference between temperature as opposed to magnetic field in these systems. And there's lots of applications that have been spurred on from this nanoscale thermometry. And I'll talk about some of the stuff that we've done in that area. And then Jean-Philippe Tétien started to then explore putting nanodiamonds onto the end of AFM probes and then building up imaging platforms to image magnetic fields, temperature and electric fields and the like. In addition to those, there's been a lot of recent work which has really extended these applications. So Peter Seigler's group in Prague have been looking at functionalizing nanodiamond particles and making them sensitive to pH, for example, and using T1 to monitor the presence of gadolinium around these particles. There's been some really nice work done in terms of virus detection and using the spin properties of the NV centers to enhance the sensitivity of these standard bioassays. Romana Schiegel's group has recently shown that we can use T1 to perform free radical detection within living cells. And so that was some work that was done recently. And I think Romana has given some nice presentation here at the Adamas presentations. And more recently, there's been some work exploring using nanodiamonds as nanoscale NMR sensors as well. So there's a lot of activity going on using these materials for a whole range of applications. And I think as we start to improve the properties of nanodiamonds more and more, the utility of these sorts of systems will grow, and the field will certainly expand into new endeavors. In terms of the nanoscale thermometry, so we started to get into this several years ago now, and one of the motivations from our perspective was to try to extend the idea of sensing using nanodiamonds to imaging. So a lot of the nanoscale thermometry applications, including some of the more recent ones, often employ a confocal microscope. And the confocal microscope will focus on a single NV center, one at a time, and then you can measure the Odmr spectrum from that single NV center, and you can monitor the temperature change that's happening from just that single NV. But often when you perform these biological experiments, you've got thousands of NVs that are incorporated into the living cell. So what we wanted to understand was, could we employ wide field imaging techniques to report from all of these nanodiamonds at once and create a temperature profile, or a temperature map, if you like, in a biological system. So we wanted to really extend the idea of using these single nanodiamond probes to imaging in wide field and reporting from thousands of nanodiamonds at once. And so we went to our favorite collaborator over at Neuroscience, and we said we had this wonderful idea. We want to probe temperature in neurons, and how amazing and fantastic this will be. And the first thing that Steve Petru said to us was that you better prove that nanodiamonds are not neurotoxic before worrying about sensing at all, which was a very important point. So there have been a lot of biological or biocompatibility studies to do with diamond, but there have been very limited neurotoxicity studies, which are actually looking at whether the presence of nanodiamond material impacts a neurological system in a significant way. And so we started to undertake that research first with Steve, before then starting to look at the idea of using wide field to do nanoscale thermometry. And so we worked with Steve's group, and in particular Emma, to start culturing neuronal networks. And so Steve's group does a lot of work where they culture neuronal networks on these multi-electrode arrays, which is what Emma is plating onto here. And then you can look at the electrical activity of this network, and then you can add nanodiamonds, for example, and see whether the presence of the nanodiamonds alters the electrical activity of the network. And that will ultimately tell you whether you've got any neurotoxicity that's happening. And so we went ahead and did that. So these are the multi-electrode arrays. This is what they look like. So you culture your neuronal network in each one of these cell plates here. Each cell plate has 12 electrodes. And so you get 12 recordings from that neuronal network on the electrical activity. And so this is what a typical measurement looks like. So you can see these spikes here are action potentials that are generated in that neuronal network. And so you've got 12 channels, and you can record. So we did recordings for 5-minute periods. And each one of these spikes is represented as a little dashed line here. So you can start to look at how the actual network as a whole is behaving. So usually when you've got neuronal networks, the network will produce a series of action potentials in bursts. So you can see how these lines are grouped together. So they collectively fire, if you like. And so you can characterize the patterns of these outputs. So these are referred to as raster plots. And then you can understand the behavior of the neuronal network on that basis. And so we performed a number of control studies at different time intervals. And then we would add nanodiamonds into that culture and then perform these electrical studies again. So we would add the nanodiamonds and perform a measurement immediately after, and then at 12, 24, and 36 hours post-addition. Now, what was fantastic to see was that there was no statistically significant difference between the electrical activity of the neuronal network before and after the addition of nanodiamonds. And this was a real surprise, even to Steve Petru, who's done a lot of work in this area. he hasn't seen a material with this kind of limited impact on the electrical dynamics of neuronal networks before. And so there are a whole range of parameters that you can characterize from those raster plots. You can look at the network burst rate, the mean firing rate, the amplitude of the action potentials, the percentage of spikes in the percentage of action potentials in the network bursts, all of these sorts of things. And you can characterize them before and after the addition of nanodiamond. And this eye plot here is showing whether there's any statistically significant difference in any of the parameters, and that will show up as a color if there's an increase in the parameter or a decrease. We looked at 28 different network parameters that are used to characterize neuronal network activity, and we didn't see any difference before or after the addition of these nanodiamonds in this system. So we were able to show from this study that these fluorescent nanodiamonds, and these were just bare fluorescent nanodiamonds, so carboxylated nanodiamonds, did not produce any neurotoxicity, which is a fantastic result. So if we want to use these sensors going forward in these sorts of systems, they appear to be biocompatible and do not exhibit any neurotoxicity, which is great. So then came the second part of the study, which was to ask the question of, can we do wide field imaging to start to report the temperature of these nanodiamonds inside the neuronal networks? And so we built this wide field imaging system, which was able to keep the neuronal culture stable at different temperatures. So basically we had a live cell imaging chamber that we would heat the whole chamber. So the objective and everything was at 37 degrees. We were able to show that employing different imaging sequences. So if we employ an image sequence where we take a fluorescence image, a standard fluorescence image, and then a fluorescence image with a microwave pulse, which is on resonance with the NV center, we can actually enhance the fluorescence image and reduce the background in that image just by taking advantage of the spin properties to do with the NV center. So we can get background free imaging, if you like, just by looking at that. And this is the exact sequence that we want to employ to do the thermometry. So if we're doing intracellular thermometry, we can do background free fluorescence imaging as well as then scanning this microwave frequency to infer the Odmr. And so we were able to show that, so here is a beautiful image of a neuron. So here's the cell body and the dendrite here, labeled with lots of nanodiamonds. And we were taking Odmr, wide field Odmr images from thousands of nanodiamonds at once. So whatever's in this field of view, we can infer an optically detected magnetic resonance spectrum from. So in this particular example, an acquisition time was about 3 seconds. So we were taking 30 milliseconds per point. And from that, we could build up these Odmr patterns, and we could monitor and follow individual nanodiamonds as the system was evolving. And so we did some proof of principle experiments at 37.3 degrees, and then we reduced the chamber to 35.2 and showed that the nanodiamonds were able to report on average that temperature change. And so this was the first time that we could start to then build up an actual temperature map or a temperature profile of that particular system. And so this was the first demonstration of multiple sensing with many or hundreds in this case of NV sensors within a living system. So there's certainly prospects for using this sort of technology in different biological systems, but I think there are a number of pros and cons to this particular technology. So in terms of the pros, diamond's a fantastic system because it's chemically inert. So when you put it into a biological system, we don't need to worry about the diamond degrading in any meaningful way. In terms of thermometry, the really important point for diamond is there is a low cross sensitivity with things like pH and redox potential. So there are lots of other nanoscale thermometers that are used in biology, but often they exhibit some sort of sensitivity to pH and redox, whereas with diamonds, we don't have that. We can readily functionalize the diamond to get them to interesting compartments and start to probe temperature temperature in different parts of the cell, which is really good. Diamond has a really high thermal conductivity, so it's able to directly report temperature changes very effectively. It's got a stable emission, which means that we can track it for a really long time. So these are all the wonderful things about diamond. Some of the real challenges are to do with the measurement speed, I think. So often in these sorts of systems, you want to track these nanodiamonds in real time and be reporting the temperature very quickly, rather than relying on averaging over multiple points. So I think measurement speed is one thing, and that's linked to the particle brightness, if you like. The other thing is that the nanodiamond systems don't report the direct temperature. So we can monitor temperature changes if we're able to follow the same nanodiamond, but we don't have a direct measurement of the temperature itself. So this often relies on an external calibration. And then I think there are a whole bunch of material properties that can be improved in terms of the brightness of the particles, the strain. So the strain has a significant impact, and the nitrogen concentration has a significant impact in terms of the Odmr spectrum itself. So here is an example of the Odmr spectrum from some of these nanodiamonds. And you can see that so there's what we think is Stark splitting from the fact that the nitrogen concentration in these materials is really high. And so you end up seeing this Stark splitting, which broadens this peak significantly. So that impacts the temperature sensitivity, for example. And you can see in when you're looking at different nanodiamonds, that the crystal field splitting position is very different for lots of different nanodiamonds. So we looked at thousands of nanodiamonds, and the crystal field splitting moves all over the place due to the fact that there's different strain in these materials as well. So that's another challenge to think about in terms of improving the utility and the sensitivity of these sorts of systems. And also the size is an interesting thing to think about. So often when we work with nanodiamonds, in the context of a cell, they're quite large. And so if you want to label them with things like mitochondria, it's often thinking about it like a big bowling ball that a mitochondria would have to carry around. And so it would be great to think about trying to get these particles smaller, brighter, so that we can start to have less impact on these biological systems. So that's where we left the intracellular thermometry work. So in terms of imaging, it's possible, but often quite challenging. And so at that point, we decided to make a bit of a pivot towards using single crystal diamond as a material platform for performing a lot of imaging applications. Now, single crystal is a little bit different. We can't get single crystal into a biological system, so we need to bring the biological system to the single crystal. And so I'll tell you about some of the work that we've been doing on these two material systems here. So before I do that, I'll just tell you quickly about how we perform magnetic microscopy using these single crystal diamonds. So now we're building these wide field microscopes that have our green excitation coming in. We shine that onto these single crystal, crystals which contain NV centers, and we image the fluorescence onto a sensitive CCD camera. And in terms of the samples that we're working with, we typically start off with a glass cover slip. We pattern a microwave resonator. Onto that, we put a thin single crystal diamond. So these are typically 50 to 100 microns thick. These are electronic grade material. And then into that, we implant a near surface layer of NV centers. So typically our implants would be 4 kilovolts, for example, which would put the NVs about 10 to 15 nanometers from the surface. So now we have this beautiful 2D array of NV centers across the surface of this chip. And then in terms of the imaging, we'll image a particular field of view in this wide field arrangement, and then we'll image that fluorescence onto a CCD camera. And so this is typically what our samples look like. So here's our microwave resonator. This is a 2 by 2 millimeter diamond that we've laser cut. And then we take our fluorescence images, and they look something like this. So they're not very exciting. There's just NVs everywhere, and we just see this blank fluorescence image. But if we take this image and then we apply different microwave frequencies in the same way as what I just talked about with the nanodiamonds, and then we take the intensity of these images, we can build up this optically detected magnetic resonance from the field of view that we're imaging here. And what's nice about this system is that we don't have to integrate the whole field of view. We can do this pixel by pixel. So we can generate an optically detected magnetic resonance spectrum pixel by pixel using this camera array. And so then if you think about adding some interesting samples, so we started off a few years ago just by cutting out a magnetic hard drive, putting that down on top of this surface, and then you get these really nice images that you can build up of the magnetic field lines from the bits, from the magnetic bits on that hard drive. So as the NVs come into resonance with the field from the magnetic bits, you can see these beautiful images as we go. So we have this really nice microscope to now start to image magnetic properties of samples that we put on top. And so I'll talk about this a little bit more in the coming slides, but we can also do exactly the same thing with the spin lattice relaxation time as well. So we can do that pixel by pixel to start to image fluctuating magnetic fields, and that will become important as we go on. So there is not many times in your being a quantum physicist that you actually get to go on a field trip to collect samples. So this was a really unique opportunity that I had to work with Jeremy Shaw. So Jeremy is an expert in these sea mollusks referred to as chitin. And so he works over at in Western Australia. He's got a beautiful lab over in Perth. And so I went to visit Jeremy after I heard about these interesting animals. So these animals have these extraordinary teeth. And so their teeth are inside here. So you can see this is their little head here. They've got these teeth. And what these sea mollusks do is they sit on rocks. And the way that they feed is that they basically eat the rock. So they mine the rock all day and they extract algae out of the rock. And that's what their main source of food is, the algae from the rock. So if you're going to sit on a rock and eat the rock, you need to have incredibly hard teeth. And so these chitin have the hardest biomineral that anyone has ever identified. And so here is what their teeth actually look like. So they have this beautiful, what's called a radula, and they have something like 73 teeth that are constantly being produced. So this is like a huge production line of teeth. And what happens is the teeth come out at the end, and these are these really hard teeth here are the ones that do the mining. They're the ones that are eating the rock all of the time. And so that's these teeth up here where they're really dark. And basically they will go through a set of teeth every 3 days. And then a new set of teeth will then come up to replace them. And what's nice about this process is you've got a whole snapshot of how this animal is producing these teeth in one radula. So you can look from the very beginning all the way through to the very end. And as I said, in terms of the hardness of these teeth, if you put this on a plot against a lot of other materials, the teeth from this animal is harder than stainless steel. And so these animals are making their own teeth in seawater. And the way that they make their teeth is they collect iron from the seawater, and they are able to assemble the iron in such a way that they can create this incredibly hard teeth that's harder than stainless steel. And they're doing it at a temperature of 15 to 17 degrees. And so if we want to create any sort of material, if we want to have something like silicon carbide, which is not that far away, we need huge plasma sources. We need lots of temperature. If the chemists try to engineer this, they need really high temperature and high acidic environments to create these sort of things. Yet this animal can do this sitting in seawater on its own. So this is a remarkable system. And there's a lot of interest in people trying to reverse engineer this system to understand how we can make materials in this way. And so if we want to reverse engineer it, we have to understand how it actually works, how the iron is formed during this process. And the really important part happens down here where there's the color changes going on. So you can see it goes from being clear to an orange and then the dark. And so the darkness comes from a material which is referred to as magnetite, which you may have heard of before. So magnetite is an iron crystal, is a form of iron, iron oxide, and it's very magnetic. And then down here, the iron is in a different form. It's in a form referred to as ferrihydrite. Now, these different forms of iron have different magnetic signatures. So ferrihydrite has a fluctuating magnetic field, whereas magnetite has a static magnetic field. So these sensing techniques that I talked to you about before in terms of Odmr and T1 can actually be applied to start to probe where the ferrihydrite is and where the magnetite is being formed at different stages. So we worked with Jeremy to get some of these teeth. We cut very thin sections of these teeth. So we cut 1 micron sections during different stages of development. So you can see this is the clear section of the teeth. This is where it starts to turn orange. And then as the teeth matures, it just accumulates more and more magnetite. That's what this dark orange here is. And so we place these sections down onto our diamond imaging chip, and you can get these beautiful, if you look at Odmr, you can get these beautiful images of the magnetic content within this teeth. So the color here refers to the presence of magnetite. So we can image where the magnetite is being formed within this teeth just by doing this Odmr imaging. And so here we're just looking at the changes in that Zeeman splitting, and that tells us about the presence of magnetite. But we can do a bunch of different forms of imaging. So we can do, so here are the bright field images. If we do Odmr, we can see that the early teeth don't show any presence of magnetite. So there is no magnetite formed at this stage of development at this particular point. So at this stage, there's lots of ferrihydrite that's going on. And if there is ferrihydrite, we should be able to see that in T1. So if we take the same images now, but using T1 relaxometry, we can now see that there is lots of ferrihydrite that's being recruited, and that ferrihydrite will then transition into magnetite. And then that magnetite will evolve later as the tooth develops. And so if we combine these images, you can actually start to see how the teeth, or how the animal is starting to assemble the teeth. So you can see that magnetite is present in this blue region here, and that is being fed by the presence of this ferrihydrite at the back and at the front of the tooth here. So this allows us to actually see how the animal is starting to assemble this exquisite material in really high resolution. The other really nice thing about using diamond is we don't have to just look at the magnitude of the magnetic field. We can actually use the fact that we've got NV centers in multiple orientations, and we can do vector microscopy. And so we can image the vector, the magnetic field vector from each of these pixels. And when we did that and we looked at different regions of the teeth, we can see that there's actually magnetic order in these teeth. And this is the first time that magnetic order has been seen at this scale over such spatial scales, when the particles during this stage of development aren't really connected. So this is a really interesting thing, because it says that although these particles aren't connected, their magnetic dipoles are oriented in the same way. And so this raises a question as to whether magnetism itself plays a role in the assembly of this material. And that's something that we're trying to understand more. Does the fact that when the iron undergoes a phase transformation from ferrihydrite to magnetite, does that happen under the presence of a magnetic field? And is it that presence of that magnetic field that makes this material so good? That's an interesting and unsolved question to come from this work. But what we were able to show is that this platform, this wide field magnetic microscopy platform, is a beautiful system to start to probe these sorts of biological problems. So the last system I thought I'd tell you about is some of the work that we've been doing with these homing pigeons here. So homing pigeons for a long time have been linked to the idea of magnetoreception. So we know that birds have a magnetic sense. What we don't know is how they're able to detect the Earth's magnetic field. And there are lots of hypotheses at the moment going around as to the precise mechanism that birds use to sense the Earth's magnetic field. there was some work done by David Dickman's group over a decade ago now, where they did some experiments where they put pigeons within controlled magnetic fields, and they were able to report their brain activity when they would stimulate various magnetic fields. So they would change the magnetic field and look at whether there was any, and look at the brain activity in certain regions. And they identified that there was a certain region of the brain that responded to that magnetic stimulus. So when there was a change in the magnetic field, there was a certain region of the brain that would light up in terms of activity. Then what they did was they thought that there might be something to do with the inner ear of the pigeon that plays a role in terms of this magnetic sense. So then they removed the inner lagina from the pigeon and then performed the same experiment again. And what they saw was that there was a reduction in the brain activity due to that same magnetic stimulus. And this provided some form of evidence that there was that the inner ear of the pigeon was responsible for some sort of magnetic sense. And so people started to then explore what was it in the inner ears of pigeons. And so David Keyes is working at the Institute of Molecular Pathology in Vienna, and his postdoc at the time, Pascal, did a lot of work looking at the inner ear of pigeons. And what they found was that within the inner ear, there are these tiny iron particles, which they termed Caticulosomes. And what was interesting about these iron particles is that there's only one of these particles per cell. So there's never a cell with two of these particles. There's only one per cell, and they're located near known sensory systems. So they're just above the hair cells on the inner ear. And we know that the hair cells are linked to sensory systems such as sound and balance and these sorts of things. And so there were the presence of these particles, but the outstanding question was, how magnetic are these particles? And are they magnetic enough to respond to changes in the Earth's magnetic field, for example? And at the time, they had no way of measuring the magnetization of these particles because they're so small. And often there are a lot of magnetic studies that were done where they chop up lots of the material and then try to characterize that in techniques such as squid, for example. But then there are lots of, those techniques are then prone to a lot of false positives. So you can collect iron oxide particles from almost anywhere. So it's really hard. It's really hard to collect a large number of these samples without getting any false positives. So we started to work with David and Pascal to understand whether diamond magnetic microscopy might be able to play a role here to determine the magnetic properties of these particles in situ, so in the biological tissue themselves. And so to give you an idea of the scale of the challenge, this is a TEM image of these beautiful particles. These particles are about 400 nanometers in diameter, and they're relatively hard to find. So there's not too many. The particles exist in 30% of the hair cells within the inner ear of the pigeon, and they're only 400 nanometers in diameter. And so we worked with David and Pascal to get some of these histological samples onto our diamond imaging chip. So here is that resonator that I told you about before, and here is a thin section of the inner ear of a pigeon. And if you zoom in, you can see, you might be able to see on your screen, there are two little black dots, one there and one there. And those two black dots are these iron caticulosomes. And so what we wanted to understand was, can we take T1 images, can we take Odmr images to start to understand whether the magnetic fields from these systems are fluctuating, static, paramagnetic, what are the magnetic properties? And so here is an SEM of one of these particles. So you can see the single cell with the iron caticulosome there. And then when we do our T1 imaging, we're able to show that we can indeed detect a fluctuating signal from one of these particles within the inner ear of a pigeon. And then we went and did a more systematic study looking at lots of different particles in the inner ears of pigeons. And we did, here we're using Odmr to actually image the stray magnetic field under the presence of various background magnetic fields. And so using this data and the data that we collected from T1, we're able to understand more about the magnetic properties of these particular iron caticulosomes. And so here are some of the measured magnetic images that we see. We see that the magnetization or the stray magnetic field grows with increasing background magnetic field. And working with Liam Hall and Rob DeGill, who was the PhD student leading this work, we were able to then simulate this scenario and simulate the magnetization of these the particles would need to have to match the measured images that we see. And what we found from this study is that these particles are not magnetic enough to be able to respond to the Earth's magnetic field. And they're 100,000 times too weak, in fact, to respond to the Earth's magnetic field. And so we think at the moment that these particles are not implicated in magnetoreception, or they're not used in this particular form for a simple compass where it would respond to the Earth's magnetic field and then open up an ion channel, for example. They're not magnetic enough to be able to do that. And so that was a really significant study that shows the power of this diamond magnetic microscopy technique for starting to look at biological systems in situ. And the really important part about this work is that it really limits the false positives. So because you can cut out the biological sample and put it directly on, you can image where that magnetic signal is within the anatomical location of the cell. So these features are a really important property of diamond magnetic microscopy going forward. So with that, I might leave it there. So I've just touched on a few of the areas that we're working on. There are a whole bunch of other interesting areas that I haven't had time to talk about today that I'd love to come back and give another lecture at another time. Here are some interesting papers from our group that we've put out recently in these various areas. And I'll finish by thanking all of the wonderful people that we work with and collaborate with here at the University of Melbourne and abroad. Thank all the funding agencies for their very kind support. And thank you very much for your attention, and happy to take any questions that you might have.
David, thank you very much. It's a very beautiful talk, very insightful, very entertaining. You not only demonstrated excellent technological developments, but also very intriguing practical applications of your magnetic microscope. So it's great work. So I have one question about these iron particles in pigeons' brains. Yep. So if they are not responsible for this navigation, as it was hypothesized initially, then what is their role might be?
Yeah, so there's two other, well, there's a number of possibilities actually. So when you look at the particles, so sometimes the particles look like this, and this is like almost a quasi-crystalline structure. And so these, or you can see it more in that image there, these individual black dots are assumed to be ferritin molecules. So these ferritin proteins, I should say. So these ferritin proteins are getting assembled into this big ball, and the ball reaches the maximum size of about 400 to 500 nanometers. And we don't see much bigger than that. And so one of the suggestions is that these are just an ion storage mechanism. So biological systems have a need to be able to store and regulate iron very precisely. So iron storage is one possible mechanism. But what's strange is that there's only one of these per cell. So if it was iron storage, it wouldn't be surprising if there was a couple of these sorts of things per cell. The other thing that people have talked about is that these may stabilize these sorts of cell systems as well. So they provide some sort of stability in terms of the flight and the balance of the bird. That's another hypothesis. And then there's a different sensing mechanism that David is looking into and others are looking into as to whether these could play a role in magnetoreception. So it's a different idea to the one where the particle just responds to the Earth's magnetic field and then there's an induced torque that happens. There's a possibility that these produce a fluctuating, well, they do produce a fluctuating magnetic field that could induce a fluctuating electric field, which could then gate different voltage regulated channels and these sorts of things. So there are a number of hypotheses that are still under investigation with these particles as to what they actually what they're doing there. And it's interesting that they're only found in birds that fly as well. In animals that fly. So if you look at rodents and these sorts of things, then they're not present in those. So it's a very interesting topic area, that's for sure.
And probably more demands for involvement of your quantum microscope as new hypotheses will appear in the field.
Yeah, exactly. And I think we've had a lot of interest from people that are looking at magnetoreception in a whole bunch of different animals and systems as well. So I think it can be quite generalized to just starting to look at the magnetic properties of small things.
Please, who has a question from the audience, please just unmute yourself and start talking and asking your questions.
There were a few questions that were, I guess, sent in that I can probably voice.
Mark, I just didn't see. Is it in chat area?
Yeah, but I think that they're sent to me, so I'll go ahead and read them.
Okay, please.
Sure.
So I guess first, it was a bit earlier in the talk. So how do you efficiently deliver microwaves to the biological sample? And I guess this is in the context of, I think, in the plate. Is it just an open wire close to the sample or some other arrangement? It's very, very high microwave power could damage the cells. So I guess, how does microwave power and delivery?
Yep, no, excellent question. So in the beginning, in our first experiment here, we had just a microwave wire, and then the cell needed to be close to that microwave wire to generate sufficient power to drive the NVs. So that was certainly one thing that we worried about, and we looked at whether there was sufficient heating that was being caused by this microwave wire. For the powers that we were working with in that study, it wasn't significantly damaging, but it was limiting in terms of the fact that the cells needed to be near a microwave wire. So that motivated the need to go to these sorts of structures, which these omega structures here. So we use these in terms of the biological studies as well. And so when we did the neuronal network work, we basically we coated this cover slip with PDMS, which meant that the salty solution that we were culturing wasn't going to short circuit our resonator here. But these resonators are really good in that we have a large, so we've got a millimeter in diameter to work with, and we can address nanodiamonds anywhere within this diameter here, and there's much less power than required for the microwave wire for us to get a decent signal. So that was one of the advances that came from these sorts of structures.
That's interesting. As a second question that came up dealt with the vector magnetometry. And it was noted that with NV centers, this is typically done either with bulk diamond or thin films, I guess, as you showed here. Is it possible to perform this with nanodiamonds?
Yes, you can. The challenge with nanodiamond is that so when we do the vector magnetometry in this case, actually, let me go back to this one. You need to, when you're doing vector magnetometry, you can do it in two different ways. You can either align a background magnetic field along each NV axis, and then you just report from the Odmr you report the ODMR along that NV axis, and then you change to the next one and look at the magnetic field projection along that axis, along that one, and then you combine that information to then convert that into a vector magnetic field. That's what we did in this particular case, because that maximizes your sensitivity, if you like, when you can align to a particular axis. When you use nanodiamonds, you don't have that luxury. You don't know what the NV axis is. If you're looking at a single
that is a real
nanodiamond,
benefit of single crystal. Yeah, that makes
yeah.
a lot of sense.
So, but having said that, we also do work where we apply random sort of background fields and look at all of the orientations at once. And so then instead of these 2 peaks, you see 8 peaks. So you get 2 peaks for each NV axis. You see 8 peaks, but typically the contrast is a little bit less. So your magnetic sensitivity is a little bit less, but it means you can do it all in one go. So that would be more like what you would do with the nanodiamond. So a nanodiamond, you don't know what the axis is, but it might be random and you might be able to resolve the 8 peaks, and then you could do it that way.
Interesting. Yeah. So those were from Saravanan, and I'm sorry how I pronounced that. There's another one that came in that I'll read out, but also anyone can unmic and ask it if you wish as well. So this is from Luis. So you actually mentioned earlier that you spent a pretty diligent amount of time measuring the cytotoxicology of the diamond itself, or of the nanodiamonds. But have you actually done any studies to act on how the act of measuring itself affects any sort of toxicology, either the microwave use or use of lasers?
No, that's an excellent question. No, so we didn't go back. So when we wanted to do the thermometry, when we wanted to do that, we needed our microscope and we needed these resonators to be able to perform the ODMR, for example. So we couldn't then take that same culture and go back and measure the electrical activity like what we do in the MEAs. Mm-hmm. So that was one of the limitations in terms of the study. So that's an excellent question as to whether the green excitation from the imaging impacts the cells in any way. The nice thing about this platform is that the power density is not as high as what you would normally do in a confocal microscope. The power density per imaging spot is less, and we're pulsing in a lot of these cases. So it's not CW excitation, so we're doing pulsed ODMR. So I think on that basis, I'd hope that there's not too much damage that's being done, but I can't categorically say that we've done those studies.
And I'll ask, I guess, a quick practical question with regards to the plate measuring. If you go back to, I guess, just the schematic, maybe. So your sample goes on top. Yeah. Yep. So diamond itself has it's a pretty high refractive index material. Does that affect the your choice of objectives or the actual imaging itself on your sample, whatever that sample might end up being?
Yeah.
Or are there any practical effects?
Yeah, no, it certainly does, and it limits. So because the refractive index is 2.4, often these high NA objectives and high magnification objectives have a very short working distance. Mm-hmm. And so when you're going through a higher index, that effectively shortens your working distance. And so if you're comparing water to diamond, it nearly halves your working distance. So that is the reason why we've had to use these thin diamond substrates. So we're working with substrates that are 50 to 100 microns thick. The typical working distance of these objectives is maybe 300 micron. So you've got to go through the cover slip, through the diamond, to then image this surface nicely. So it does impact it in terms of we're limited to about 100 microns of these diamond chips. So typically, so what we do is we get these diamonds cut and polished. So we'll take a typical 300 or 500 micron diamond, cut it down the middle, polish both sides, and then use them, implant them, and then use them for imaging. So yeah, it certainly does have implications. Yeah, if they could find an objective which is suitable for a 2.4 index, that would be phenomenal.
Yeah, I think that'd be rather hard to do, but maybe possible. I mean,
Yep.
it would actually be an interesting. It'd help a lot.
It would, absolutely. Yep.
David, I have another question.
Sure.
Can you please describe an ideal diamond plate with shallow NV centers from your perspective? What you would like to have as a working instrument? Sure.
So, one of the key challenges is turning the nitrogen into useful NV centers. And so, when you start getting very close to the surface, the conversion efficiency from N to NV changes a lot. And so, typical studies will show that you've got a conversion efficiency much less than 1%, for example. And so that typically means that we're putting more nitrogen into the material, which is then impacting the coherence times and the sensitivity of these experiments. So what is of interest to us and some of the things that we're working on at the moment is to try to improve that conversion efficiency. And trying to understand what limits that conversion efficiency, what are the processes that we need to kind of avoid. And then if we can do that, then the implantation doses can come down. And then I think a lot of the magnetic sensing and electric field sensing becomes even more sensitive, if that makes sense. So at the moment, we're implanting at a density of 10 to the 13 per centimeter squared. And that, when you convert that into effective nitrogen concentration within that small delta layer, it's equivalent to about 100 ppm. So it's just it's really, really high. It's equivalent to the HPHT material. So that's something that we'd like to see come down. So if we can start working with chips that have effective densities around the 10s to 20 part per million that are still bright enough for us to be able to image and do this work, that would be fantastic.
And what say depths about 10 to 20 nanometers that you consider, or you don't need so shallow for your application, since your objects are kind of on their own quite large, right? Quite thick. Yep. So probably you don't need those super shallow NVs, and they can be at higher depths and higher quality.
I think it depends on the application. So for ODMR, a lot of the time the sample will be large enough that you can afford, and the fall off is consistent with 1/R. And so that means that you're not losing a lot of the signal over the depth of the implant. So you can afford, if you're doing ODMR imaging, you can afford to be a little bit deeper. When you're doing T1 imaging, that relies on the exchange of magnetic dipoles, and that has a dependence that goes on 1/R to the 6. So T1 is extremely sensitive to the standoff between the NV layer and the sample that you're interested in. So the closer the implant can be to the surface for T1 sensing, the better. And so that's where we would win a lot in terms of our sensitivity to measuring fluctuating magnetic fields. But the challenge is, once you bring them really close to the surface, how do you keep them in the negatively charged state for long enough and maintain their coherence properties? How do you functionalize the surface to reduce all of the magnetic noise and electric noise? So yeah, it's a real sort of, so it depends on the application, and it's a bit of a trade-off between coming closer, but then being noisier in terms of the surface and things like that.
In terms of patterning of NV location, is there any benefits to have patterned location versus just random as it comes from the implantation?
So pattern, do you mean?
If you would do masking during nitrogen implantation, you would have some, just say, arrays of NV produced rather than these randomly distributed NV centers.
Yeah, so I think, yeah, patterning is really good when you're trying to understand how like the signal would change with different implantation parameters and things like that. For us, we've found that these 4 keV implants at 10 to the 13 provide decent T1 and Odmr properties to image. So then our preference is to make the whole chip sensitive. So then whatever sample we put on top, we can image anywhere in that sample and it's going to be the same. And so I think the patterning is really good to identify what is the best parameters for an implant. And then once we know what that parameter is, then we would typically then decorate a whole sample so that we could use that whole sample for imaging.
So say for imaging protocol itself, it's not needed, right? I mean, not of advantage to have kind of arrays of FNDs. Yeah,
no, it's certainly not needed. And the uniformity of these implants is pretty good. So you can go anywhere on this diamond and it will look the same. With the parameters that we have at the moment. So which is a nice thing when you put a biological sample down, you don't know where the interesting part is going to be. So yeah, you need to make sure that you've got sensors everywhere, I guess.
So David, actually we have another question, biological question from the audience from Luis. What sort of temperature sensitivities could you achieve with ODMR? And is that sensitivity good enough to measure any naturally occurring temperature fluctuations in the biological system?
Yeah, again, that's a really good question, and I think it's a bit controversial at the moment. So the temperature sensitivity that we had was about a degree, I think, in this work using this technique, and we were kind of limited, as I said, to the width of this ODMR, which limits the slope here that we're imaging or using to infer temperature. Whether 1 degree sensitivity is sufficient to detect a temperature change is an open question in my mind. So there are lots of debates in the literature at the moment about what sort of temperature changes occur within a single cell and whether there are temperature gradients that exist and whether these gradients are much higher than that. There was a paper that reported that mitochondria can get up to 50 degrees in temperature, which would be extraordinary if that was the case. So I think, when you're looking at large temperature changes, if that was actually true, then this probe would be ideal and it would be sufficient to detect that. When you're looking for small temperature changes, I think we do need to do work around the temperature sensitivity. I think we'd like to be much better than a degree or half a degree. We can push that down by an order of magnitude through better properties, brighter particles, these sorts of things. That would be an interesting place to be. And then we could start to ask these questions in a realistic manner. A lot of the demonstrations that have happened so far have been where the laser has induced some sort of heating. So it's not naturally occurring temperature changes within a living cell. It's either been heating of gold nanoparticles in the case of the Lucan group. This more recent work in Science Advances was using laser heating to affect changes. There's been different changes in embryo genesis through laser-induced heating as well. So these are all artificial mechanisms to create temperature changes. And we did the same thing through just changing the temperature of our microscope by 2 degrees. So I haven't yet seen, so it'd be interesting to know if Luis is aware whether there are reports of intracellular changes of that scale naturally.
Yeah, I'm yet to come across it, naturally, but would be very interested if we could find a system that could work with the sensitivity of only 1 Kelvin per square root hertz. Until we find a system that can do that, I think we need to wait until the diamonds have better properties and can read out at a better sensitivity.
Yep, completely agree.
So it's quite a prolonged discussion after your talk.
Oh, that's good.
A lot of questions, which is always great for a lecturer. Do we have more questions? So it seems that you satisfied the curiosity, current curiosity, and people will continue to think about all that beautiful work and future perspectives that you showed in your presentation today. So David, thank you so much. It was a very exciting presentation, and on behalf of the audience, I would like to thank you. And I'm sure a lot of people will watch a recorded video, as it often happens after the presentation.
No worries. It was an absolute pleasure, Olga, and I hope to see many of you at the MRS meeting in Hawaii.
Yeah, see you in Hawaii in a week.
That would be great. All right, thanks very much.
Mhm.